Hi, my name is Joerin, and I am presenting the work G6PD deficiency and X chromosome ancestry patterns in Brazil. First of all, it's important to say that glucose 6-phosphate dehydrogenase deficiency, or G6PDD, is an X chromosome linked red blood cell enzymopathy that causes neonatal jaundice and acute hemolytic anemia triggered by certain foods, certain medications, and infections. It is related to the important G6PD cellular function that is critical for protecting red blood cells from oxidative damage. In humans, the gene G6PD is located near the telomeric region of the X chromosome, and more than 200 mutations are well described in exonic regions. However, intronic and other variants outside the G6PD gene have been also associated with deficiency but are still poorly studied. The G6PD deficiency prevalence correlates with geographical distribution of malaria and has been associated to protection against infection. It is particularly prevalent in Africa, Asia, the Mediterranean, and the Middle East. It's important also to say that the Brazilian population has an important African ancestry contribution, and the Amazonian Brazilian region presents a high risk of malaria. Other regions, like the Central West, the Southeast, and the South, are not endemic but present residual transmission of malaria due to the proximity to malaria-endemic regions. Also, aminoquinoline-derived antimalarial drugs are recommended for curing and preventing malaria relapse, but they can induce severe hemolytic anemia in deficient individuals. In addition, chloroquine and hydroxychloroquine have been widely promoted by Brazilian official authorities and professionals as treatment for coronavirus disease, based in limited clinical data. Considering that, the aims of this work were to analyze the prevalence of variants associated to G6PD deficiency in the G6PD gene and outside its open reading frame in Brazilian populations from 4 different regions and according to ancestry and phenotype. To do that, we analyzed a sample from the north, the central west, the southeast, and the south of Brazil, genotyped by the Illumina genome-wide microarray, including sample of heteroclassified blacks, whites, and self-declared Amerindians. Data analysis was performed on 27 single nucleotide variants from the G6PD gene and over 100 SNPs from the X chromosome for ancestry analysis. We used the software Plink, Admixture, the R packages Pegas and Stats, and the software Arlecchino. We studied 27 G6PD related variants. Some of them are missense well-studied variants, such as the exclusive from Portugal Miradouro, Betica, and Santa Maria. Other missense variants were the Seattle and the Mediterranean, and also the African A and A-. As a result, we observed that there is a strong overlapping between phenotypic categories of admixed Brazilians when considering X chromosome ancestry, whereas self-declared Amerindians presented a predominant Amerindian ancestry. All the variants studied were in Hardy-Weinberg equilibrium in the 3 groups analyzed. We also observed that African variants presented higher frequency among Black Brazilians, including the missense variant G6PD-A. Among males, G6PD-A- variant, which determines moderate deficiency, showed similar frequency in blacks and whites, but over 90% of its carriers had over 15% X chromosome African ancestry. We also detected the allele RS1050-757C, which has been associated to G6PD deficiency in the 3 groups studied. All the A- males were from the Central West. In addition, we also detected that over 7% of the Central Western males and over 5% of the Southeastern females carried the African A variant, which determines mild G6PD deficiency. The variant Miradouro was detected in 1 white individual from the Southeast. We also analyzed molecular diversity indices in male haplotypes from the 3 groups, and we observed that the highest diversity indices were in Black Brazilians, whereas the lowest were for Amerindians. Thanks for your attention.